Hi, we're continuing with life science and cell biology in a nutshell. We just learned about translation and how ribosomes control translation. Ribosomes are uh, the region of the cell where uh, mRNA sequences are turned into proteins. mRNA is a one-stranded chain which comes out of the nucleus because it is transcribed off of the original DNA sequence. Uh, so a gene on the DNA or section of the DNA um, is turned into a single strand of mRNA, and the mRNA comes out, goes onto the ribosomal units, and is turned into proteins. We learned that the difference between DNA and RNA is this, that DNA is double-stranded while RNA is single-stranded, and RNA has an extra oxygen. It's called ribose, is the sugar in RNA, while deoxyribose, which is missing an oxygen, is the sugar in DNA. And also, RNA has the uses uracil as its one of its four bases, and the other three bases are cytosine, adenine, and guanine. So uracil instead of thymine, which is in DNA, the difference between thymine and uracil is that thymine has two extra hydrogens, and uracil is missing the two hydrogens. So, other than that, the chains are exactly the same of mRNA and DNA. So, sections of the DNA are transcribed. The different sections of the DNA, the sequences determine which sections get transcribed. The beginning parts of the sequences to be transcribed are called promoter regions. They bind a protein called transcription factors, which start the transcription of the gene. And the RNA polymerase is the protein that helps with the polymerization of the mRNA. So the sequences that need to be transcribed are transcribed off the genes, and these sequences which leave the nucleus are turned into proteins. So, of course, various proteins may help with the transcription process, with starting and stopping of transcription, and making sure the mRNA is in the shape that it needs to look like before it leaves the nucleus. So excess intron sections of mRNA, which are non-coding regions, may be removed from the sequence. So only the needed part of the sequence, which is going to be turned into proteins that are the coding regions of that molecule, mRNA, will be uh, the sequence or chain that comes out into the ribosome and is turned into proteins using our tRNA molecules as helper. tRNA brings amino acids. The anticodon region of tRNA binds the codon regions of mRNA, and each anticodon is codes is bringing a different amino acid, and the polypeptide chain grows. And there may be multiple ribosomes making multiple polypeptide chains, and different and proteins help with this process that are called chaperone proteins, with the helping of um, just making sure that the chains are um, made correctly, the polypeptides. This is what it looks like in the microscope. The next organelle that is in the cell is on the exterior of the nucleus. It's a network of membranous tubules within the cytoplasm of eukaryotic cells that is continuous with the nuclear membrane called the endoplasmic reticulum. Usually has ribosomes attached and is involved in protein and lipid synthesis. It forms a series of flattened sacs or cisternae within the cytoplasm of eukaryotic cells and serves multiple functions, being important particularly in synthesis, folding, modification, and translocation and eventual transport of proteins. The membranous sheets and tubules that begin near the nucleus and extend across the cell are what make up the endoplasmic reticulum. Rough endoplasmic reticulum is involved in protein production, protein folding, quality control, and dispatch. The endoplasmic reticulum's membrane typically constitutes more than half of the total membrane of an average animal cell. The RER is contiguous with the nuclear membrane. RER is called rough because it is studied on its outer surface, the surface in contact with the cytosol with ribosomes. So this is what it looks like. The nuclear pore, which where molecules like mRNA molecules come out of, and they bind to the ribosomes on the RER. The cisternae are the folds, and that's where protein production occurs on the ribosomal unit. Membrane-bound ribosomes are firmly attached to the outer cytosolic side of the ER. About 13 million ribosomes are present in the rough endoplasmic reticulum in the average liver cell, for example. Rough ER is found throughout the cell, but the density is higher near the nucleus and the Golgi apparatus. So we'll learn about the Golgi in a bit. This is where molecules are getting ready to be exported from the cell itself. The Golgi. So ribosomes, membrane-bound, are responsible for assembly of proteins. So and once they are assembled in the ER, the Golgi is where it has the vesicles which transport them out of the cell. So translation occurs on the ribosomes, which are on the ER. So proteins are made. Certain cells of the pancreas and digestive tract produce a high volume of proteins as enzymes. Many proteins are produced in quantity in cells of pancreas and digestive tract. Function as digestive enzymes. Rough ER, working with membrane-bound ribosomes, takes polypeptide and amino acids from cytosol, continues protein assembly, so including at an early stage, recognizing a destination label attached to each of the proteins. Proteins are produced for the plasma membrane. The Golgi apparatus, secretory vesicles, plant vacuoles, lysosomes, endosomes, and the ER itself. Some of the proteins are delivered into the lumen or space inside the ER, whilst others are processed within the ER itself. Thank you.